Yeah, welcome to this video. So respiration is split into 4 sections, and this is the first section. Stage 1 is called glycolysis. So if we break down the meaning of the word, first of all, so the glyco refers to some kind of carbohydrate, so sugar. Carbohydrates are made of sugars, and lysis means to split something. Okay, so it literally means splitting sugar. So the sugar that we're going to split is glucose. Okay, and the sugar is glucose. So what we're going to do is we're going to break that carbon down. Now, glucose, you might remember from the GCSE equation, is C6H12O6. So it's a relatively big molecule, look, made of all of these 6 carbons, 12 hydrogens, 6 oxygens, and we've got to break that molecule down because it's very big. Now, glucose contains a lot of energy. If we broke down glucose all in one go, it would release so much energy it would actually kill the cell. So what we do is we break it down in little stages, releasing a little bit of ATP at a time, and that amount of ATP is a perfect amount of energy for the cell to do individual tasks. So glycolysis is splitting glucose. Now, you'll probably recognise glycolysis starting off with glucose, something like this, going down, splitting into 2, and they go down, and they make 2 molecules of pyruvate. Okay, so what we'll do is we'll have a look at how this process happens, what the stages are called, how we explain them, what language we use. If glucose is C6H12O6, then it obviously has 6 carbons. And because we split it in half, the pyruvates that we make are going to have 3 carbons each. Okay, so we're splitting in glycolysis a 6 carbon molecule into 2, 3. So remember, it means splitting sugar. Okay, the sugar that we're going to split is glucose, and we're going to split the glucose from one 6 carbon molecule into 2 molecules that are 3 carbons each. So let's go from the top. So we start off with glucose. Try and make it a little bit smaller to fit more on the page. So what happens first of all is that the glucose gets phosphorylated. Now, because the glucose has 6 carbons, we often draw those carbons, or we can draw those carbons as circles like this. So that represents that it's a 6 carbon molecule. Okay. Now, the first stage that happens is that we do something called phosphorylation. Now, phosphorylation means adding a phosphate group to something. So what we end up with after phosphorylation of glucose is a molecule called phosphorylated glucose. Okay, so rather than just glucose, we've then got a molecule called phosphorylated glucose. Now that phosphate has to come from somewhere. So we draw it as our 6 carbon molecule still, and we can draw it with the little phosphates attached. Okay, but they have to have come from somewhere. And the place they come from is from the ATP molecules. So if we have an ATP molecule just here, adenosine triphosphate, tri means that it contains 3 phosphates. And we could simplify it like this. So we could draw adenosine triphosphate like this, showing that it's got a molecule with 3 phosphates attached onto it. Now what happens is if we remove one of those phosphates, if I rub that off, if one of those phosphates is removed and I'm just left with 2 phosphates, it's now become a molecule called adenosine diphosphate. Di means 2, so it did have 3 phosphates and now it's got 2 phosphates. Now the action of removing that phosphate from there releases some energy, and also it obviously releases that phosphate. So if we break down ATP into ADP, I'll just rub those off for now. So if I break down ATP into ADP, by drawing this curved by drawing that curved arrow just there, what I'm showing is that that phosphate that's come off there has actually been given to this reaction. So this was adenosine triphosphate. This arrow represents one of those phosphates being removed and being passed to this arrow. So we've got glucose coming in from the top here, we've got ATP coming in here, and where they meet, the phosphate is being transferred from the ATP onto the glucose. And if we follow the arrow down, we're making a phosphorylated glucose. So you can see that one phosphate sticks on here, and one phosphate has stuck on here. Okay, so by starting with glucose coming in, ATP coming in, we make ADP and phosphorylated glucose. Now you can obviously see that there's actually 2 just here, so we need to just make it add up. So 2 ATP creates 2 ADP, all right. And then the 2 phosphates have stuck onto the glucose to produce phosphorylated glucose. So we could say an ATP molecule is hydrolyzed. Hydrolyzed means broken apart. And a phosphate group is released. The phosphate group attaches to the carbon 6 of a glucose molecule to produce glucose 6-phosphate. The enzyme hexokinase catalyzes this reaction. Glucose 6-phosphate then turns into fructose 6-phosphate, and the enzyme glucose phosphate isomerase catalyzes this reaction. Another ATP molecule is then hydrolyzed, and the phosphate group that's released attaches onto carbon 1 of fructose 6-phosphate, and this becomes fructose 1,6 biphosphate. The enzyme phosphofructokinase catalyzes this reaction. So you can see that you could break this down into a series of 3 smaller steps, but this will be fine. This summary here will be fine. I was reading that from your student book. If you want to go into that detail and you want to see what it says in there, it's just in your student book. Okay, but the most important thing is that from glucose to phosphorylated glucose, we're using 2 ATPs to donate phosphates onto the glucose molecule. Now, the reason why we add phosphates onto the glucose molecule is because we want the glucose molecule to split. Okay, so this phosphorylated version is much less stable and it splits into 2 smaller 3-carbon molecules. So let me go on to a new board. So at the end of that stage that we've just talked through, we've got phosphorylated glucose. So that's our starting point. You can obviously just keep going down the page. You don't have to keep going onto a separate diagram. So phosphorylated glucose. So this molecule is going to split into 2. Okay, so remember how many carbons it had? 1, 2, 3, 4, 5, 6. Remember, it had a phosphate on there and it had a phosphate on there, and it's going to split. So it's obviously going to split into 2 3s. Okay, the phosphates will still be attached on there. And the molecules that it splits into are 2 3-carbon sugars. Okay, now this process here, you can just say that a 6-carbon molecule splits into 2 3-carbon molecules. If you want names for those molecules, we usually call them triose phosphate. Same again. Okay, you could abbreviate that to TP, triose phosphate. Okay, so the 6-carbon molecule of phosphorylated glucose splits into 2 3-carbon molecules that are still phosphorylated. Okay, then we've got the bit where we're going to go down. So you remember we started with glucose, we went down, then we split, and then we go down again. Yeah. So we've got stage 1, which we did on the first slide, stage 2, which we just talked about the splitting, and then we're going to do stage 3, the next 2 downwards parts. So let's start off with our 2 molecules of triose phosphate. Remember how many carbons each of those has. So 1, 2, 3 with a phosphate attached, 1, 2, 3 with a phosphate attached. Okay, now we're going to keep those as 3-carbon molecules, but they're going to lose their phosphates. Okay, and the molecules that they make are called pyruvate. But we need to know a series of steps that happen to get from triose phosphate to pyruvate. So we're going to go down to pyruvate. Pyruvate is still a 3-carbon molecule, but it doesn't have the phosphate attached. So how do we turn a molecule of triose phosphate into a molecule of pyruvate? So what happens is that we're going to move some hydrogens around. So the molecule we're going to use for this is called NAD. So NAD is what we call a coenzyme or an electron acceptor or a hydrogen carrier because it carries hydrogens around. Hydrogens contain 1 electron, so you can call them electron acceptors or hydrogen carriers kind of interchangeably. But what happens is if we start with an NAD molecule, what it can do is it can pick up a hydrogen and they become what we call reduced NAD. Now you could call reduced NAD NADH, and it actually kind of makes more sense to call it NADH because it picks up a hydrogen. Okay, so triose phosphate donates hydrogens to NAD to produce reduced NAD or to produce NADH. Okay, now what you'll find as we go through respiration is that NAD is almost like a little hydrogen taxi. It carries hydrogen from the early stages of respiration to the later stages of respiration. So NAD is a coenzyme. You can call it an electron carrier, but its job in respiration is to carry hydrogen atoms to stage 4. So here, this NAD is picking up a hydrogen that it gets from the TP, and it's going to take that hydrogen to stage 4. Okay, now, if you remember from GCSE, we had this little way of remembering 2 types of reaction, oil rig. So the O was oxidation is loss, and the R was reduction is gain. So here, what's happened is that TP, in this little point where the lines kind of touch, TP has given hydrogen to NAD. Okay, so if the TP has given hydrogen away, it's lost hydrogen, so it's been oxidized. So at this point here, we could say TP has been oxidized. At exactly that same moment, NAD picks up the hydrogen And so it's gained hydrogen, so the NAD has been reduced. Okay, now that reaction that's happened just there is what we call a redox reaction. RED, because the NAD was reduced, OX, because the TP was oxidized. So anytime a hydrogen is passed from one place to another, there's a redox reaction because one of the molecules has given the hydrogen away and one of them has gained it. So one's been oxidized and one been reduced at exactly the same moment. Okay. So this process here is going to happen on both sides. So from this TP molecule, NAD will pick up a hydrogen from TP to produce a molecule called reduced NAD or NADH. All right, so same thing on that side. It's just given it hydrogen. The second thing that happens here is that we're going to make some ATP molecules. So if we want to make ATP molecules, we need to start with ADP and add a phosphate. So if we start with this time ADP, adenosine diphosphate, and we want to make ATP, okay, so what we can do is we can use this phosphate here. This one's got two phosphates. If this phosphate here is given to that ADP, it will now have three phosphates. So it's become ATP. So what's being given across there is a phosphate. Okay. Now this is one of the places where it doesn't quite add up in terms of the diagram because we actually have two ADPs plus phosphates making two ATPs. Now we're only getting one of those phosphates from the TP molecule. The other inorganic phosphate is just being added in, okay, from elsewhere in the cell. But it is 2 and 2 there in terms of your numbers. So we do the same on this side as well. So two ADP molecules have phosphates added to them. We always say inorganic phosphates. When we write phosphate, like I've written there, really I should write a little tiny I next to it for inorganic phosphate. That's just traditionally how we write it, a capital P and a little tiny subscript I at the bottom. So two ADP molecules produce two, ATP molecules there. Okay. And then that produces our pyruvate molecules, which are the first step of the next stage. So if we go back to the first one, just to recap, we started with glucose, and in this stage here, we actually used up two ATPs. So that stage there, if we just write it in the corner here, we're actually on minus 2 ATP on that stage. On the middle stage of glycolysis, here, oh sorry, where's the middle stage? That's it, the one where it splits. There was no mention of ATP on that one, okay, so no ATPs involved there. And then the third part of glycolysis, this one here, we've made two ATPs from that triphosphate and two from this one. So in this stage, we've made plus 4 ATP. So to summarise that again, just get a new board. We started with glucose. It went down, then it split, then it went down. And at the end, we ended up with 2 pyruvate. Up here, we had minus 2 ATP. We used 2 ATPs. And down here, we made 4 ATP. So what we can say is that the net overall is plus 2 ATPs from glycolysis. Okay, so overall, glycolysis has produced 2 ATPs, which is good, but it's not a lot of ATP. Respiration in total makes about 38 ATPs, so it's not that significant, but it is important that we know it. The other thing it's made that's very, very important is these reduced NADs. So these reduced NADs here are going to carry the hydrogens to stage 4. Remember, NADs are a coenzyme, an electron carrier, and their job is to carry hydrogen atoms to stage 4. So we've also made 2 reduced NADs. Okay, so we've also made 2 reduced NAD molecules. So this is really important because this is the outcome of glycolysis. So glycolysis is the first stage of respiration. Okay, there's more detail in your student book if you want to look at it, but this will be fine. Make sure you know that we start with glucose, that we end with pyruvate. Make sure you know how many ATPs are made. Make sure you know that it makes reduced NADs that are carrying the hydrogens to stage 4. The other thing that we haven't mentioned so far is that this stage happens in the cytoplasm. Okay, so it's strange because we always learn that respiration happens in the mitochondria, but this first stage here actually happens in the cytoplasm, not in the mitochondria. Okay, and that's the end of that video.